Okay. Hi, everyone, and welcome to our June 24th edition of the Rutgers Ornamental IPM program webinar. We're going to be talking about some current issues and topics for integrated pest management, and got a good program for you. So I'm Bill Errickson. I'm the agriculture agent for Rutgers Cooperative Extension of Monmouth County, and I'm also joined by Steve Rettke, our IPM program associate, Tim Waller, our ag agent in Cumberland County, and Erin Quinn, our IPM program support staff. And so we're going to be talking about some materials today in our controls. We just want to remind everyone to always follow the label, and that's the law. And things may differ from state to state. I know some folks are viewing this regionally, and that's great. So just make sure you follow your own state's regulations. And what we're talking about is specific to commercial operators in New Jersey. Okay, so right now, if you look at the bottom right corner, we've got our growing degree days in northern New Jersey. We're at about 980. Central New Jersey is at about 1130, and southern New Jersey is at about 1225. And we're also in the second day of a 3-day heat wave. It's about 98 degrees right here in central New Jersey right now. So those growing degree days are going to be increasing very rapidly now at this part of the season. So we're going to talk about today a new beneficial, some parasitoid wasps, leafhoppers, fall webworms, Japanese beetles. Steve Rettke is going to talk about midsummer leaf drop and oak lacebug and warm season spider mites and mealybugs. And then Tim is going to get into phytotoxicity diagnostics, some sanitation updates, and how we're monitoring our scale crawlers. So we've got a good program, and we'll go ahead and get started. So some of the updates. We also recently launched our website. So if you haven't checked that out, that's available. It's live and online. All these webinars are being posted to the website after they're recorded and presented. And these resources, as we go throughout the season, we're continuing to build them into the website. So check that out. And then also we have the free updates that come out through the Rutgers Plant and Pest Advisory. So anytime we're dealing with pest issues in real time, and updates or upcoming programs, those go out through the Plant and Pest Advisory. So we've been talking about beneficials and leading off the program each time talking about that. And these are some beneficials that I observed in the field over the last week. On the left, we see a predatory mite on a silverleaf hydrangea. That's Hydrangea radiata. And you can see they're a little bit larger than spider mites, for example, and they're also very fast. So when I was observing this mite, he was moving around really rapidly on the hydrangea. And then on the right, you see two hoverflies on the American elderberry that we're looking at here. We talked about the hoverflies last session. And if you can see in the top right portion of that image, you actually can see also a lady beetle larva in the background on the leaf. So multiple beneficials, that we're starting to see. And when you observe those, in the nursery or in the landscape, we want to preserve their habitat and protect them, and maybe monitor them and adjust our spray programs accordingly, because they can provide a lot of assistance for controlling pests. So for this session, the beneficials we want to focus on are the parasitoid wasps and flies. And so these are smaller beneficials that will actually lay their eggs into pest species. And so they can provide great biological control. They're host specific, so certain parasitoids are going to colonize specific host species. And in the picture here, we're looking at aphids, as we can observe the aphids, and then we see the mummified bodies of aphids where a parasitoid wasp has laid its eggs, and then those eggs have hatched and exited the body of the mummified aphid. And you see that exit hole on the aphid there. And so that's a good indication that parasitoids are present in the environment. And you may see low aphid populations that are at manageable levels. If you start to see those mummified aphids, that's a good indication that we may want to protect those beneficials that are in the area. So you want to look for the swelling bodies of the aphids, those mummified, and then the exit holes. That's a good indication. Even if you're not observing the parasitoids directly, that's an idea that they're around. So we can see the picture on the left there. You see the tannish brown aphid bodies. Those are the mummified aphids. So those have been had their eggs laid in them by a parasitoid wasp. And then we can see stuck to a yellow sticky card in the middle picture on the top. And that's a parasitoid wasp. So you see it's a little bit smaller. It's got that constricted waist. And there's many different species of these parasitoids. On the bottom, that's a horntail parasitoid wasp. And on the right, we see parasitoid wasps looking for eggs of spongy moth to lay their eggs. So that's a spongy moth egg mass, and the parasitoids are inspecting that egg mass to actually lay their eggs into the spongy moths. So many different shapes and forms of the parasitoids. So having a diverse habitat of flowers that bloom throughout the season, lots of native species, they're going to encourage these parasitoids to be present. And we want to, adjust our spray programs to, incorporate the presence of parasitoids and other beneficials. So just be careful with broad spectrum contact materials, or if we're using systemics and it can get into the pollen that the parasitoids might be feeding on. So many of those materials will also have restrictions for in-bloom applications, and that's why. So is that we don't want that to get into the food source for these beneficials to have these non-target effects. So just kind of being conscious of that and modifying our programs to allow these beneficials to be present and to help us with that pest control. Okay, so our first pest, something that we do not want to encourage is the potato leafhopper. And so these should be out and all throughout the state. So they start to come out at 784 growing degree days, and they have 4 to 5 generations per year. So this is kind of like a season-long pest with multiple generations. And they have piercing, sucking mouth parts that can actually release a toxin, and that restricts the movement in the vascular tissues of the plant. So in some ways, we call that hopper burn generally, and in some ways that can look like drought stress, and it's because there's this toxin that's causing this response in the plant. And so potato leafhoppers and other leafhopper insects can also transmit other pathogens like bacteria or phytoplasmas or viruses. They can be vectors for these pathogens that can get into the plants and cause other problems as well. So one of the characteristic movements of potato leafhoppers is they have this sideways movement to them. So if you wave your hands over the plants and they start to move and they're going sideways, that's a good indication that we're looking at potato leafhoppers. And we see these on a lot of different species, maples in particular, oak, birch, ash. So if you're growing trees, keep an eye out for potato leafhoppers. We'll show you some hopper burn pictures here we see in the container nursery. We've got on maple, you see that looking like a little bit of drought stress, but that's that burn on the edges from the restricted vascular movement and the toxin in their saliva. We see how it expresses on oak as well, another maple on the bottom, and likely a hollyhock on the right side. One thing I should also mention about potato leafhoppers is that they do not overwinter in New Jersey. So that's a good thing for us, is that our winters will actually kill them. But they move up from the south each year. And you can see the picture on the bottom left, their movement coming up with the jet stream, air currents, the warm air coming up and bringing them up from the south. So our natural predators can help with potato leafhoppers. So our lady beetle larvae, lacewing larvae, minute pirate bugs, beneficial insectary plants that can support those populations will help in the nursery or in the landscape. And monitoring for leafhoppers can help as well. So one thing we can do is to use yellow sticky cards that we do for monitoring lots of different insects. And if you're growing maples, that's one of their favorite host plants. So if you put yellow sticky cards out around the maples, that can give you a good indication that the potato leafhoppers might be present. You may also have higher populations if you're close to agricultural fields. That they can be hosts for potato leafhoppers also, and this may bring them into the nursery. So just keep an eye out, especially if you have neighbors who are growing other crops, or if you produce other crops on the farm in addition to the nursery crops. So there are contact materials. Our pyrethroids will work, but those are broad spectrum, so you just want to keep that in mind. There are some systemics also. But then some of our biorational materials like neem oil, or the active ingredient in neem is azadirachtin, that can also work pretty well for potato leafhoppers. Okay, next up we've got the fall webworm. And so previously we had talked about the eastern tent caterpillars, and fall webworm can look quite similar, but it's a little bit different for how it behaves on our plants. And so the fall webworms, they're going to make their webs at the tips of the branches, and they stay within that protective webbing. And as they want to continue to feed, they can create more webbing, and then they'll spread slowly that way. And so they kind of extend their web and stay under it, but it's kind of a thin web. And so this helps for control, because it keeps them kind of concentrated within that web. And we can prune them out. If it's concentrated in that way, we can prune them out if we find them early, or we can treat them appropriately, spot treatments. They will attack many different species of trees. Over 100 different species of trees can be hosts for fall webworms. Ash, elm, linden, sweet gum, willows, walnuts, oaks, hickory, fruit trees, all these different species can be affected. So you see some severe infestation on the left. The tree is totally covered by fall webworms. We do not want things to get to that level. But then on the top there, got a weeping cherry, and you can see it's just one branch there. And so you could prune that out, or you could open up those webs and expose them for birds to be able to consume them. But they're going to stay in that web, and we can identify them that way. They can also either have a black head or a red head. So it's actually the same species of insect, but it's a different race. So can look out for those. And again, they're going to attack many different species of trees. Even ginkgo, you can find them on, and there's not many pests that will bother ginkgo, but fall webworm may go for it. So this is actually a native insect. So there's a lot of natural predators that have evolved with it. So parasitoids, if we're encouraging their populations, they can help to keep fall webworm in check. Birds can consume them also, so if you can break up those webs or spray them with a jet of water, that can affect them also, or just pruning them out. Our contact materials, pyrethroids and carbamates, those will work, but again, being broad spectrum, we can have non-target effects, so be careful with that. There are some systemics that can work also, but only if it's a really severe infestation, or if you have really high populations in the past, you may not need that. BT, or Bacillus thuringiensis, that's a good biorational material, right? That's the bacteria that can affect caterpillars, and it gets into their gut and kind of dissolves them from the inside. That's good if you can get it on early, and it'll affect those younger instars. The spinosins, those can be also effective as a reduced risk material. So another common pest that we see this time of year, every year, probably starting to see it in central and southern New Jersey. They're starting to appear, maybe hasn't shown up in northern New Jersey yet, but this is the Japanese beetle. And we talked about oriental beetles in a previous session. And if you remember, with the oriental beetles, the adults were not doing that much damage. It was more the grubs underground that are feeding on roots. And that's not necessarily the case with Japanese beetles. And so with the Japanese beetles, we're going to have the adults and the grubs both causing damage. And so the adults are coming out, June through August to feed, and they're actually attracted to plant feeding damage. And that's why we start to see them cluster like that and have that concentrated damage. It's like once that starts, they're attracted to that, and then more and more can come and continue to cause that damage. So their life cycle, they have one generation per year, but it's a long life cycle because the grubs can cause damage underground also. So the adults will feed, will come out midsummer and feed, lay their eggs in the soil, and then the larvae, the grubs, are going to start to feed on the roots. So these can affect many, many different species, in particular roses, grapes, linden, and birch trees. And then we're going to see the life cycle here. So right in the middle is when the adults emerge, but for a lot of their life cycle, they're actually underground as the grubs. And the grubs are going to feed on the roots in spring and in the fall, with the adults coming out in the summer, and then the grubs go a little bit deeper in the soil in the winter. So depending on your control measures, you're going to have different control measures for an above ground adult insect versus a below ground grub feeding on the roots. And here we can see more of that damage, total skeletonizing of the leaf, as we see on the left. It's more damage in the center. We've got the rose being chewed up on the right, raspberries, and we see the white grub, how they look when they're underground in the bottom center there. So this should be a fairly familiar pest to most folks. But there are a lot of natural predators. Their birds will feed on Japanese beetles, and you may see increased bird activity around your plants, and that could be an indication that Japanese beetles are present. There are some mammals and other beneficial insects, predatory and parasitoid insects that will feed on them. The adults are going to be a little bit slower on cool, rainy days, so that could be a good time for hand picking or knocking them off, or more physical controls. And some of the plants that are a little bit less susceptible would be boxwood, dogwood, magnolias. So if these are real concerns for you, or if you have a client that they're really concerned for, you may want to look at some of those other plants. I know a lot of folks will go for kind of broad spectrum materials like the carbamates or the pyrethroids when Japanese beetles come out, just to get them under control, because they can be so damaging. But just be aware that, if you have other non-target organisms present, as we have a lot of beneficials around this time of year, that they could be effective. Some of the systemics can also be affected here, or effective against Japanese beetles. And then we have biorational materials also. The milky spore bacteria, that's can be effective, but kind of over a multi-year process. And that's to target the grubs and to try to reduce their populations, to reduce the adults. So that can be kind of a long-term way to affect them in the surrounding environment. Neem oil as a directant, the active ingredient in that can also work for Japanese beetles. All right, I'm going to turn it over to you, Steve.
Okay, thank you. Welcome back, everyone. By the way, some of you may not realize, but we've now reached the halfway point with these webinars. We've scheduled 12 for the year, and so this is number 6. So those of you who have attended all 6, you probably should receive some kind of a reward. But I think the only thing we can say is thank you. So anyway, yeah, I'm going to talk about a lacebug species, 3 spider mite species, also a scale species. But I'm going to start off with an abiotic problem that we find starting about this time of year often. And there's some deciduous trees in particular, and they are the linden tree, the birch tree, and the sycamore tree, that are really susceptible to this mid-season or midsummer leaf drop. Those are species of trees that during the springtime, they grow a lot of excessive amount of leaves, more than they really need for photosynthetic capabilities. And so because of the fact that there's so much plentiful water during the springtime, and so they do have more leaves than they need. And though we get more of a drier period, as we're starting to develop now with the hotter temperatures, I've already seen it, not on my great birch yet, but I've seen it at some other locations where you may have 30, 35% or more percentage of the leaves dropping off. And that creates concern to people. I've had nursery growers over the many years, a handful of them ask me, what's going on with my birch or my linden or so forth? And I say, well, I think it's okay. They think perhaps there's a root system problem or maybe a wood borer. But after I investigate that and I don't see anything, I will say, well, it kind of looks like you just have a mid-season leaf drop. And because of the fact that these leaves are, such excessive amount, and they do, have a lot of evapotranspiration occurring this time of the year, the plant wants to reduce that moisture, water loss, and so they just simply drop many of the leaves that they don't really need. And so, yeah, this the key thing is, and I'm sure there's other species, but the ones I've seen most often and a consistent basis are the linden, the birch, and the sycamore. Go to the next slide, please. And so this is actually my backyard, and this is a gray birch defoliation with excessive leaf growth. It's because of the fact that, as I said, when water was readily available in the spring, they were really able to grow a lot of excess foliage. And so, all right, so let's move down to some of these pests. And the one I'll talk about first is a lacebug pest. It's in the summer, we see it. And it's one of the Corythuca, which means that it feeds on deciduous plants. And the oak lacebug is the one I see a lot of. And they have certainly been occurring already. They've been laying eggs. They overwinter as adults, and they come out usually in June out of their overwintering hiding places and bark crevices and so forth. I see them most often on white oaks in particular, but there are other deciduous trees they'll feed on too, but white oaks are certainly the key one. They do have those rectangular shaped wings as shown there. And what's curious about them is that when they start laying eggs, typically around the second week of June, at least in central New Jersey, they have a egg pattern that often has been stated they look like a miniature Stonehenge, or maybe a group of bowling pins all stood up, and maybe a 3/4 inch diameter circle with the female lays the eggs, and they insert them, in an upright pattern, and they insert them into the foliage, and they start hatching from that area. And so there's 3 to 4 generations a year, and the first generation of the oak lacebug doesn't really amount to much. The stippling damage is still relatively mild, and it doesn't draw much attention. But as you get into especially by the time you get to the 3rd generation, or even at the end of the year, the 4th generation, the amount of stippling damage becomes far more, pronounced. But even with that, I think it's often stated that oak lacebugs, they may have bark, but they really don't have much bite to them, and it's primarily an aesthetic damage that they're producing. And this I wanted to show you these eggs right there, and that's in the upper left. You see the Stonehenge look of the eggs, like bowling pins, or you might add, and you see the ones to the left of that. Those are first instars that just hatched out, and then you have an adult that's tending her eggs. And I think she's already probably laid most of her eggs. And then going to the right of that, you see a lot of more signs and symptoms. You see all the, oh, by the way, I should mention the fact that lacebugs are not going to ever be found feeding on the upper parts of the leaves. They're going to be only on the lower part. And that's the case with all lacebug species, no matter if it's deciduous or evergreen, broadleaf evergreen. And so that's a good close-up of the eggs, again, from Utah Cooperative Extension. And then you see the mild symptoms that are showing there. You might, by this time, perhaps start to see the symptoms that look like this. Hardly even draws any attention, but then that aesthetic damage will increase as you get to the 2nd, 3rd, and finally the 4th generation. And so a big white oak tree there that's just only about maybe 400 yards from where I'm speaking right now. It's a huge, maybe a 200-year-old white oak, at least 200 years old. And every year it gets oak lacebugs, but it never seems to amount to anything. It doesn't really cause much damage for someone, a landscaper, for example. I mean, this is not a nursery situation, of course, but for someone to spray, dozens and dozens of gallons of an insecticide on a tree like that would be just kind of almost a crime, unless it's maybe really a biorational type material. But it's often not necessary to do these treatments. And I kind of showed this slide maybe a few weeks ago, just showing you where these insects are feeding. Now I'm going to talk about spider mites later on, but both the oak lacebug and the 3 spider mites that I'll be speaking on, they're going to be feeding in the palisade layer, that layer of cells that is just under the upper epidermis, where the sunlight is going to be concentrated the greatest. And so the most amount of photosynthates are going to be concentrated there. And so that's why the spider mites and the oak lacebug like to feed there. The spider mites and all those species are feeding only on the undersides of the plant, with a major exception, which I'll talk about in a minute, and that's the oak spider mite. They feed on the tops. And then I'll talk about a scale as well later, another more unique type of a family of scales, and it's a mealybug, which is actually a scale, and they feed in the phloem, as shown there on this diagram. So, right. And as far as controls, you know, they have these contacts that will work. You know, you have carbaryl and pyrethroids, you got the neonicotinoids, they all work well. The spinosad, and you got some biorational materials there. Acephate is kind of an old organophosphate, but it has a translaminar property, which makes it kind of good to use, especially if you're going to be spraying the tops of the leaves and you got these underneath leaf feeders. You know, the translaminar, it'll be moved into the leaves, and so it'll negatively affect the pests that are feeding, even though they're not hitting them with contact directly. And then you have some of the biorationals, and soaps and oils can work as well. I mean, I think it's usually probably best to use them against the immatures, and I got a few pictures of those. I think maybe I've already showed them. So, all right, so the one that I had there, the fluorantraniliprole, that is a reduced risk material. It doesn't even have a caution label on it, and so it's one of the more benign materials that we have available to us, and that's actually a true systemic as well, and it's a biorational. Bulgaria bassiana, well, I mean, if you're going to use a material like that, you're going to have to anticipate probably spraying 4 or 5 times before you're getting satisfactory efficacy. All right, so let's go to some of these mites now, and these are the oak spider mites. They overwinter as eggs. Okay, you see the first generation is occurring now in central New Jersey. It's occurring basically everywhere. And so they have, several generations, probably at least a half dozen or more generations throughout the entire season. And so they come out as eggs on the bark, and they begin to feed, and they start laying eggs. And it's another one. All these mites, by the way, are all warm season spider mites, and they all thrive in dry conditions. I think you all know that pretty well. And what's kind of curious about this particular species, though, and this Oligonicus is the genus it feeds in, but the genus that it is. But the fact that the oak spider mites feed primarily on the upper side of the leaves, and I always thought that to be rather, interesting. The fact that why are they feeding on the underside? Are they on top of the leaves and not the undersides of the leaves where they have more protection? Well, that's kind of a mystery to me, because you think they'd be much more exposed to, wind and rain and so forth, and maybe even more readily apparent to predators. But anyway, they seem to have evolved to do that, and so that's something that's kind of unique about them. And the eggs and also all laid on the upper leaf surfaces, as shown there, that photograph. Those are caskins, the white caskins. You may have some white eggshells. When you see white like that on leaves, it's usually talking about caskins, the exoskeletons, as the mites molt, like they're not insects, but they molt just like insects do. And then the caskins are going to be white, as well as the eggshells are going to be white as well. And so, but they're all just feeding on the upper surface. You can see the stippling present there, and just little minute chlorotic feeding that they produce. And then what happens if they have a high enough population, these little minute feeding areas, they start to coalesce, and they can appear to be much worse as they get higher populations. And then finally, the final generation will lay eggs on the woody tissue to overwinter again. And this showing you, this happens to be a sawtooth oak at the upper left, and you see that's the upper leaf surface there. You see caskins and eggshells and stippling damage. You turn that leaf over, where you normally would expect to find things like lace bugs and other types of spider mites, but no, not with the oak spider mites. That they're clean. They're not on the undersides. So it really makes it curious. And then you see the symptoms there in the center, and then close up on the right, you notice how they seem to have an accumulation of eggs along the veins, and then the eggs hatch. The caskins are also there as they begin to molt, and they spread out, and you can see the stippling occurring there as well. But you'll notice them, the top of the leaves, you'll see them along that the leaf areas, the lateral veins, and they kind of concentrate there with the caskins and their eggshells. Okay. as controls, and I tell you, I think I'm going to probably continue to go with the same slide for the next two mites, and just to save time. But there's just a whole bunch of good miticide materials that are out there for us. It was a good number of years ago, maybe a couple decades ago, where we were losing all of our miticides. And over the last 20 years or even more, we have produced a lot of unique chemistries, these biorationals, these mite growth regulators, even though they're not necessarily brand new, some of them are more newer than others. And so there's also these reduced risk materials shown there under the systemics, bifenazate and asaquinazole. They're all reduced risk, and they have different modes of action, which are kind of interesting, but I don't have the time to talk about them. And then the mite growth regulators are always good. But again, remember with the mite growth regulators, you don't really prefer to use those when you have severe populations of mites, because you're not going to be killing the adults. You will kill the larvae or the nymphs. They hatch out at first as larvae, they have 6 legs, and they become nymphs, but they got the 8 legs. But those are immatures, and that's when the IGRs work well against them, because they negatively affect their molting capabilities. And so that can be useful for smaller populations. But when you get a lot of adults, even though the eggs will more than likely be sterile, you're not going to get much control. It's going to take a long time. It may take 10 days or more before you get these populations under control if you're using IGRs or mite growth regulators. Okay. Let's move on to maple spider mites. This is also a little bit interesting because there's not really anything at least I couldn't find anything as far as the growing degree days. And apparently this is somewhat variable. And I looked at a number of different fact sheets from various universities. None of them had growing degree days. And so apparently this is one of these pests that we haven't established a good, reliable growing degree day range for them. But they do overwinter as both adults as well as eggs, and they're going to be on the bark and little bark crevices, and maybe in the crotches of the twigs and so forth. You see the eggs and the adults that are overwintering. They do migrate as they begin to emerge and move out the foliage. As leaves come out, you'll see them on the undersides of the leaves. And this is kind of an aesthetic problem. Again, they're not really causing a great deal of damage to the trees, and I indicate here that they're going to be mostly on maple trees. In fact, it's only indicating maple trees there. This is one that's mostly host specific. There might be a couple other hosts that weren't listed there, but I can't think of anything offhand. And so, again, these are mites that are feeding in the mesophyll, that palisade parenchyma cells, and so you're going to see that stippling taking place. And there's many generations that occurs each season. So. Right. And so you see the symptoms there in the upper left, pretty benign for the most part. And then you barely see some visible active mites. This is what the naked eye would look like without any kind of a hand lens. That's why hand lenses could be so valuable to use and actually see the mites. I always say I never walk the nursery or a landscape without a hand lens. That's really a critical tool. But perhaps you can just barely make out along these veins, the primary vein and the lateral veins there. You can see a little bit of spots, these dark areas. That's where the maple spider mites are at. And the interesting thing I heard in a nursery a few years ago, you notice there's a lay of trees that extends for maybe 250 yards or so. And there's dozens and dozens of these allays. It's a huge nursery that is in Cumberland County. And the one on the left are oak species, and the one on the right are maple species. Now I indicate that they're host specific. So unless they're oak mites, they're not going to feed on the oaks. And these are maple spider mites. And so those oaks are clean. They have no symptoms at all. But look at the symptoms on the maples. And so my suspicions were that at this nursery, most nurseries, they really want to have as few years as possible before they can start sowing their trees. And so they do provide a lot of nitrogen for them. And so, the mites like the nitrogen, and so it kind of builds up. Nitrogen is a limiting element in the environment. And so the mites really do prefer that nitrogen to increase their reproductive rates. And so it's just amazing the symptoms that were all the way down 200 yards or more of these maple species trees that were there. And, you notice that they were all staked, and that's the case with all nursery. in the field. We have these larger trees, and the canopy gets a little bit top heavy. And you in the branches, the main stems, they're not really sufficiently rigid yet. And so you got to support them. Otherwise, you get some wind coming in. You're going to be snapping a lot of your trees, and so they avoid that with the, of course, with the stakes. All right, so I'm not going to go through these because I know I'm going to be running out of time. And so this is showing you, these are something you can go back to and look at the various materials. And it's mostly the same ones each time. And so for that reason, I mean, there's a lot of good materials there. And a lot of them are reduced risk or biorational types. So just don't immediately go to the easy stuff like the pyrethroids and just think, you know, you're going to get maybe resurgence with those. And so, you know, when you really get a heavy population, perhaps you kind of have to resort to that. But that's why we monitor, and that's why we try to catch these populations early, so we don't have to go with the harsh materials that create, you know, those negative impacts. And so the last mite would be the honey locust spider mites, and they overwinter as adults, and they're classified as a warm season mite. You can see how early the eggs are hatching, though. And so even though I'm talking about them now, they've actually been active for a while, but they do reach their peak in the summer times. And they, again, with a mite, you're going to have multiple generations, and they kind of build up as, you know, I guess I'm not quite sure how many generations, but I would assume more than a half dozen or more as you go through the season, and sometimes perhaps put more than that. And so you go to the next slide. And by the way, before you do that, go back real quick, Bill. No, one more. Okay, can you go back? And I'm not running out of time. You see the circle there, the red circle. Those are larvae of mites. You know, as I indicated maybe just previously, I said that mites hatch, and they're larvae at first, and they only have 6 legs. And then, because we all know mites have 8 legs, right? No, but the larvae only have 6. And then they molt again, and then usually there's 2 molting stages where they're nymphs. I believe that's correct, 2 nymphal molts. And then they have the 8 legs. And so I just wanted to show that to you. And so they're showing you another one at the upper left where you got the congregation of eggshells as well as caskins, usually at the base of the needles there on the honey locust. The leaflets, by the way, that's a compound leaf. And so that's where they're kind of congregating. And then showing there at the bottom left, you have the overwintering adults and the bark crevices and so forth, and the bud scar locations. And you can look for these during the, you know, before the leaves come out, and you can get an idea. You might want to use a hand lens to really help you, but even with that, you know, the red might stand out. You can pick them up with the naked eye. And you can maybe do some dormant oils treatments. If you have a history of them on your honey locust, that seems to build up worse and worse each year. You can see the damage that they do there on the right. And they do can create a lot of damage and defoliation as they build up over the summer. So, and I'm going to move through this. That's more the same with the materials and so forth. And so my last pest I want to talk about are the Taxus mealybugs. Okay, and yes, they are scales as indicated there. They're a family of scales. They're not armored scales. They're not soft scales. They're not felt scales. And so their own family of scales, and that's the mealybugs. And Taxus mealybug is one of the species. I see them most often on hues or taxus, and of course rhododendrons as well. And they're going to be found on a few other trees also. And some of them, as you see there, are deciduous. So they overwinter, interesting, as first instar nymphs, whereas our soft scales, if you remember, overwinter as second instar nymphs. Okay, and they're going to be on the branches and the stems. And so the thing about mealybugs, though, is they are difficult to control. They have this powdery, waxy covering that they put over themselves that really gives them pretty good protection against some of our materials that we try to control them with. And so this is one of the tougher ones. And I see mealybugs in greenhouses. And if it's just maybe a handful of plants, what the growers will often do in a greenhouse, they'll just toss those plants because they know that if they attempt to control them with sprays, they may get decent suppression or they may not. And so they often just forget about those plants. If there's not too many of them, they'll just toss them before they start spreading around. But go to the next slide, and you can see the damage that they can do. The Taxus mealybug, they have these pairs of white tufts and usually about 15 to 17 of them on each side of the bodies. And then with the Taxus mealybug, that last pair is maybe about a quarter or a third the size, the length of the actual mealybug. And that's a good way to identify them. There's a few other species. There's like striped mealybugs, and there's long-tail mealybugs, and there's a few others as well. And they will feed only on the branches, by the way, not feeding on leaves. As you can see them down deep down on the stems there on the left, you see they have to look for them on the stems. And then look at the damage they did on this rhododendron on the right, where you had an infested plant. And there was a point where that plant probably needed to be discarded, and I know it was. And just showing the comparison with the on the left, where it hasn't yet, there's no infestation there. And so that gets to the point where these mealybugs can create a lot of damage, and these are the Taxus mealybugs in particular. All right, so anything different about these? Now we're talking about scales now, so maybe a little bit different. Just to kind of emphasize that one thing I wanted to say is that apparently the neonicotinoids are not, as you might suspect, they're phloem feeders, and so you might think the neonicotinoids would work well, but apparently they're just not always that effective. And so that's one you have to be aware of. There is another neonicotinoid that's listed as well. That's acetamiprid, which is a reduced risk material. It's a spray only, and I've heard that maybe works a little bit better than the other neonicotinoids. And then you got soaps and oils that you can try, and then some of those insect growth regulators that you know, they're molting type hormone disruptions, the pyriproxyfen and the bupropion, and you know, they're expensive, but there's a little bit of a sticker price with those, but they do go a long way. Just a little dab will do you, and they can really last for a long time. You don't have to use too much when you apply them. Okay, let me put it to an end there.
All right. Well, hey, everyone. Thanks again for being with us. Hoping everybody's getting a little bit of a temperature reprieve from the day. So we're going to talk a little bit about phytotoxicity through the lens of what would be a better named slide is probably something of the effect of what are we looking at here? This is one of the main things that I get calls about. Y'all don't save the easy ones for me. And we start to see symptoms that don't really fall into categories that we are familiar with a classic insect issue or a classic disease issue. Whether we're looking at interveinal chlorosis, we can have the appearance like mottling that would raise the question, are we dealing with root diseases? Are we dealing with viruses? We will start to have instances of contorted growth or discoloration plus contorted growth, where we're really starting to question what is going on here. I very much wish I could give you a one-to-one on what you're seeing, what is always causing that issue. But a better way to approach this is just some questions that everyone should be asking themselves when we are approaching this idea, are we dealing with phytotoxicity issues? So there's a lot of words here, but I kind of made this in a way that hopefully this helps some folks kind of work through what might be happening in a particular situation. So we all do tank mixes, but it's worth asking the question, was that material used in a tank mix prior? Did we try out that tank mix in all the different crops where we might be seeing this phytotoxicity issue? If the answer is no, we haven't tried this out before, that's when we'd start to look at that particular tank mix. Have any herbicides been applied? Herbicides make plants do funny things. Did we apply herbicides? Were they over the top? Could this be the culprit that's causing that type of damage? Something that I've seen many places throughout the state is asking the question, are we near an agricultural field? Particularly soybean row crops, where they could be using dicamba, glyphosate, etc. Are we potentially downwind of the prevailing wind direction, and we might have gotten some drift? Are you the operator using cleanup herbicides around your houses? It's a very common practice to keep weeds down around our high tunnels. Was it really hot outside? And the reason that that's important, a day like today, this would be one of the worst times that you could apply something like glyphosate. Some of the formulations can volatilize, and they kind of drift around like cigar smoke, where there's not a very easily discernible pattern of where that material could be getting onto plants. And if you're having damages and they're also on a thin bark tree, again, this is where I'd really start to look at glyphosate damage. It can cause damage that won't become evident for maybe a year or two, but it's absolutely something that can happen. Did you apply a new pesticide? Is that potentially causing the issue? Did you put it into a tank mix to replace something that you're normally mixing? And again, has that tank mix been trialed in other plants? If it hasn't been trialed, then that may be a culprit as well. And then just keeping an eye out for some obvious damages. One of the images earlier, immediately my brain went to, we have a leaf that's very cupped in appearance. That's a classic symptom of dicamba damage. So just kind of having an idea of this pesticide-related phytotoxicity is important. However, usually that's just a piece of the puzzle. Are we looking at a pest issue? Have you searched up and down those plants? Did you find a huge population of aphids? Do you have leaf curl and you want to look deeper and maybe you have found a bunch of things like thrips inside of there? Do you have wounds on the plant? All of these could be related to the symptoms that we are seeing. Do we have obvious signs of viruses? Or if we are thinking maybe this is fungal related, do we have pycnidia or the presence of spores? What happens if we incubate those leaves? And again, going back to the idea of if we saw insects there, if we had thrips, aphids, leafhoppers, etc., we may be dealing with a situation where we're having these insects vectoring a disease. Environmental, all of our plants, I mean, today is a great example of it. Today is a harsh day for our plants. Are they being overwatered? Are they underwatered? Are they sitting in water? I'm going to talk about sitting in water in a little bit here as well. But all these are really important. The other big thing with a day like today, and I saw this a lot last year, where the only rational outcome that we could get to was fertilizer dumping, where the fertilizer was put in one location in the container, got excess of 100 degrees, incredibly warm temperatures, and those little pearls break down very quickly and you have a rapid release that'll boost the salts. We're getting into toxicity events where we're going to burn our root systems and potentially lead to discoloration in our plants. And then just noting that the shade the shade requirements of those plants, and they could also be involved as well. Wind damage is another one that I've seen that could look like phytotoxicity or other issues, environmental issues that we've had. All of this is to say is that diagnosing phytotoxicity issues is quite complicated, but these are kind of the first place where you're going to start taking a stab at what could be causing this problem. And that's also a plug here for a plant diagnostic lab. They might not be able to tell you exactly what phytotoxicity component was involved. They can do residue analysis for different chemical pesticide applications. But moreover, they can tell us that we are not dealing with a virus or we are not dealing with a fungus. And in a diagnostic approach, knowing what you are not dealing with is just as valuable as knowing what you are dealing with when we're kind of trying to solve this kind of moving target of a problem. All right, so the next thing I want to talk about in timely fashion is spring sanitate, or after this big spring push, we've got our plants out. We're shuttling around plants. We're throwing away material. We may have instances where we have open houses where we can do some really good work at limiting the number of diseases, bugs in general, and weeds as well in those houses. So really preparation now, we are preparing ourselves for the whole next season until this time next year. So if we have a house that is full of debris, there's a great opportunity for pathogens to go throughout the season, over winter, and become a problem again next spring. So just kind of putting it into perspective here, when we are looking at this could be in the field, this could be probably more applicable to in a high tunnel situation in our hoop houses, is that diseases can overwinter all over the place. Weed mats are a great place for things to persist, and especially in our leaf litter. Particularly if we have a crop that we had a problem with and it's dropping leaves, or we're ripping up plants to see what's going on with their root systems, there is the opportunity there for pathogens to persist for longer periods in those locations. So if we can get them out of the system, the better off we're going to be. So just to kind of put things in perspective, this leaf here, we see these little tiny black dots. These are pycnidia. Within each one of those tiny little black dots, there could be hundreds to thousands of spores. So removing this single leaf out of the equation is greatly going to help safeguard our plants moving forward. So the cleaner we can get it, the better off we're going to be moving forward. Some of the really persistent diseases, and this kind of looks like the list of plant pathogens that I interact with regularly, particularly our oomycetes. They have all kinds of different survival structures. Many of our leaf spots, our mildews, and a number of the big hitters when it comes to causing real problem, particularly in containerized nursery. It's also important to note if you're doing your own propagation, a lot of the diseases that are listed here and others can be transmissible from the mother stock. So paying close attention to the plants that you are propagating off of can be really valuable as well. And it's much better to discard plants early on than to have population explosions, whether that's a disease or a pest that could be coming in. So, you know, I'm very much a realistic person. I know what it's like. Throughout the season, our houses get dirty. It's all a matter of going through and cleaning them up afterwards. But the more sanitation that we can do up front, the better off we're going to be. And this very much could lead to a lot of issues that we might be seeing early next spring that could have gotten their foothold this time of year last year. Had we gone through and really cleaned up the place, we might not have been dealing with it in a year's time. So one of the things I'm really interested in researching is how long pathogens can persist in our weed mats and how long they're going to stay present there. I do know if our houses are constantly full of debris, they're constantly full of algae, and we're just rotating plants on top of them, we are going to have more problems than we would have if we had cleaned up after it. So two other points that I really wanted to touch on here. So when it comes to sanitation, early on this season, there were a number of verticillium tangential problems that I worked with a couple nurseries that were utilizing the pot and pot system. It can be a great system, but if we are not sanitizing, we are not cleaning our pots up, or socket pots rather, we may be putting plants right back into a disease-conducive environment. This is particularly important with a lot of root diseases like verticillium, fusarium, rhizoctonia, things like phytophthora, pythium, etc. If we have a contaminated socket pot and we, at any point in time, we have water that floods up through that system, maybe something happens with our drainage, that's the exact situation that we can lead to inoculating the plants that we are trying to sell. The other thing too is make sure that you're at least sanitizing. Preferably you are changing out your emitters if you're dealing with diseased plants. It's not a horrible idea either to completely change out the socket pot if we have something like verticillium or something like phytophthora that is impacting our plants in that location. So avoid standing water. Just to kind of put things in perspective, so I work with phytophthora and root rots. The way that we inoculate plants and make sure that they get diseases is that we give them wet feet. We have them sit in standing water. When it all comes down to it, many of our root diseases, water is critically important for that. I know that during production season, fixing locations that may have standing water is not the highest thing on your priority list. But if you do have an open house, and you can do something to prevent that water flooding in that location moving forward, now is an excellent time to do that. So once we've got our plants out of there, you find yourself with an open house. If we can, the way I would approach this is we're going to physically remove any of the debris. Hopefully you're not blowing leaf litter from the house that's open into a house that's full of plants that you're going to be selling in a little bit, but we're trying to remove that material the best we can. Remember that one leaf has all those spores that could cause problems for us. So the cleaner we can be, the better off we'll be. If you can inject sanitizers through your irrigation system, this is going to clean out potential biofilm formation inside of your irrigation lines. It's the kind of the first step in cleaning up your emitters, and the spray off of that could help us, will help us to some extent clean up our weed mats. And we're cleaning that material out, and then we're using a chemical sterilant to help clean us up. If you're applying any type of pesticides, herbicides in that house, you're doing that in a clean house, or you're knocking down weeds around it. And once you've got everything situated, that house is clean, you're bringing plants back in. If at all possible, you should be growing different plants than you were growing them the year prior or the season prior. I know that's not always possible, but that is a foundational element of plant disease management, and that's crop rotation. And anything that could get algae or nostoc, anything that gets slimy in our operations are the exact locations where a lot of our diseases are able to persist for very, very long periods of time. I know I'm a very realistic person and I know what that can mean, but if you can clean and sanitize regularly, you're going to be better off. That's just a fact of the matter. And just being cognizant that you aren't putting irrigation things on the floor or on the weed mat or inside of a greenhouse for that matter, if they're not meant to be something like a drip irrigation system as well. All right, and then the last thing I just wanted to touch on, and this is an area of this program that we're going to be constantly building on, and that's monitoring for scale insect crawlers. This is a great way to help us know when crawlers are emerging, so we can utilize the correct spray programs, hopefully with softer chemistries. But it is also a way for us to gauge how effective our sprays have been, particularly in situations that have multiple generations. So we've got sticky tapes shown here. We've got double-sided electrical tape. Basically, you wrap the correct way across a branch, and then you flip the tape around and wrap it the other way, so the scale crawlers have to move across it, and you can visually see what's happening. That being said, using a hand lens is incredibly important. Scale crawlers are very small, but if there is a crawler hatch, they're going to be pretty numerous, and you're going to be seeing whether it's orange to a pinkish to a purplish, very, very small organism in mass. You're probably looking at scale crawlers. Some other things to keep an eye out for are the presence of predators. Things like if you're seeing sooty mold on a leaf and you're also seeing something like a lady beetle. Here we've got a twice stab ladybird beetle. That's giving us a great indication that there are potentially soft scales in the area, aphids in the area, etc. Like we've stated before too, if we see a lot of ants on a plant, those ants are probably farming some organism that is producing honeydew. So basically, observation is what's so important when it comes to scale crawler monitoring. You're not going to be able to check every plant, but if you had a number of them set up in your operation that you're going to stop at and you're going to look at regularly, it's going to save you a lot of money in getting control of a pest. And it's also going to save a lot in potentially wasting sprays when they weren't really giving us the most bang for our buck. So with that, I just want to say thank you very much for attending again. Halfway through this, at this point in time too, moving forward, if you have topics that you'd like us to talk about. So we are hitting a point in time where we're kind of through the first wave of the insects. There are ones that we're going to keep talking about, But if there are topics like the sanitation or phytotoxicity, something like that, if there are topics you'd like us to cover, we're all ears and we'll do our best to accommodate. So we're gonna jump into this material next week. And I believe that is the last slide that we have.